Welcome to the lecture series in food microbiology. This is in continuation with the discussions we have been having on foodborne infections. We discussed about Shigella, Salmonella, Listeria, etc. and the infections which are caused by them when you eat or consume food or water contaminated with these. Today we are going to discuss about some more of these infections. Vibrio gastroenteritis. Historically, cholera has been one of the most feared diseases by the mankind. It has been endemic to the Indian subcontinent, where it is estimated to have killed more than 20 million people in this century. And during the 19th century, there were a number of pandemics of Asiatic cholera spreading from the Indian subcontinent throughout Europe and the Americas. Pacini in 1854 is credited with the first description of the etiological agent of cholera when he observed large numbers of the curved bacilli in clinical specimens from cholera patients in Florence. Not how generally accepted because of the widespread occurrence of similar but harmless vibrios in the environment. See, he saw it in 1854 and he said that it was these curved bacilli which caused the cholera pandemic. But his view was not accepted generally because you see a lot of curved bacilli in the environment. So how do you pinpoint that it is this which causes the particular disease? And Robert Koch firmly established the causal link between Vibrio cholerae and cholera when working in Egypt in 1886. The Vibrio bacteria generally live in coastal waters. Around 12 species of Vibrio can cause a gastrointestinal illness in humans. Cholera is only one of the gastroenteritis or enteric diseases caused by Vibrio species. You have other species of Vibria which are capable. Among the genera Vibria itself, you have multiple species which are capable of causing human infections. Vibrio cholerae is one among them. And these Vibrio generally live in coastal waters. The illness produced by Vibrio bacteria is generally called as vibriosis. In most of the cases, the illness results from eating contaminated food like raw or undercooked shellfish from water that contains the bacteria. Exposing a wound to contaminated water can cause a Vibrio infection on the skin. Symptoms and signs resemble those of the food poisoning, and these include watery diarrhea, abdominal cramping, nausea, vomiting, fever and chills. And avoiding raw shellfish is one step towards the prevention of vibriosis. Specific treatment is not required in most of the cases because of water loss, because of diarrhea or vomiting. Replacement of fluids would be required. Most people with a mild case of vibriosis recover after about 3 days without permanent effects. And an infection with the species Vibrio vulnificus can cause severe life-threatening illness. In the genus Vibrio, 4 species have been implicated in foodborne illnesses: the Vibrio cholerae 01 and the non-010 groups, Vibrio mimicus, Vibrio parahemolyticus and Vibrio vulnificus. These are the 4 major species which have been implicated and which are the causes of foodborne illnesses among the genus Vibrio. The Vibrio parahemolyticus gastroenteritis. The gastroenteritis caused by this species, this is very common, and this accounts for 40 to 70% of the total bacterial foodborne disease. And the high incidence of this gastroenteritis is directly related to the consumption of raw seafood. As we saw, Vibrios are coastal water dwellers. So when you consume seafood obtained from such coastal waters, or naturally the seafood like your mussels, scallops, whatever is the type of seafood you are talking about, that would be contaminated with this bacteria. And partially cooked or raw seafood consumption from such area would definitely implicate foodborne illnesses. The cells are gram-negative, non-sporulating, motile, curved rods. And the 4 biochemical identification, they are catalase and oxidase positive. The strains grow in a medium containing glucose without producing gas, but they are unable to ferment lactose and sucrose. So these are the biochemical markers we look for in the laboratory for identifying Vibrio parahemolyticus. And not all strains of Vibrio parahemolyticus are pathogenic. The foodborne pathogenic strains can cause hemolysis because the presence of heat stable hemolysin, and this is designated as the Kanagawa positive. At present, the 23 kilodalton heat stable hemolysin, called as the thermostable direct hemolysin or TDH, this is considered to be the toxin which causes hemolysis of the foodborne pathogenic strain. All the pathogenic strains of Vibrio were found to adhere to human fetal intestinal cells in the cell cultures. The toxin production rate and its level are directly related to the cell growth, the cell concentration, and pH of the environment. And if the toxin forms in a food, heating does not destroy it, it is heat stable. So, when the pathogenic strains were allowed to infect intestinal cells grown in the lab in cell culture, the production rate of the toxin and was directly related to the rate of the cell growth, the cell concentration which was there in the cell culture medium, and also to the pH of the medium where the cell was being grown. When you consider this aspect, and if you see that this is a foodborne illness, if the toxin is present in the food, normal heating will not destroy because this is a heat stable toxin. In unrefrigerated, raw and cooked seafoods, Vibrio parahemolyticus can grow rapidly, especially at 20s to 30 degrees centigrade. So, raw seafood as well as cooked seafood, which is unrefrigerated. And suppose you have cooked seafood but it has been exposed to parahemolyticus, then if the food is stored outside in a temperature of 20 to 30 degrees, the Vibrio parahemolyticus can grow in it. And in temperature abused seafoods, where at the time of storage the temperature is fluctuating without being fixed, the cells can reach an infective dose very rapidly, even from a low initial population. And once the pathogenic strains grow to infective numbers, even heat treatment cannot destroy the toxin. So, if the bacteria was present in the food and it was stored, and during storage there was temperature abuse, naturally the organisms will replicate, multiply. And once these reach from the lower number to the infective dose which is present, then they will start secreting the toxins, and even heat treatment will not be able to destroy these toxins. Vibrio vulnificus septicemia. Vibrio vulnificus infection is the leading cause of death related to seafood consumption. This virulent gram-negative bacterium causes two distinct syndromes. The first is an overwhelming primary septicemia caused by consuming raw or undercooked seafood, particularly raw oysters. The second is a necrotizing wound infection acquired when an open wound is exposed to warm seawater in high concentrations of Vibrio vulnificus. So, two kinds of septicemia are caused with Vibrio vulnificus. One is primary septicemia, which happens when you consume raw or undercooked seafood. The second is, see, you have a wound and you happen to swim in coastal area, or you get exposed to seawater containing Vibrio vulnificus. Then the wound gets exposed to the organism, and then and that organism will infect the wound, causing the necrotizing wound. So, this is a picture which shows us the wound and the septicemia which is caused by Vibrio vulnificus. Following consumption of the contaminated seafood, in this case many raw oysters, the cells penetrate the intestinal wall and produce primary septicemia in 20 to 40 hours. The symptoms of this primary septicemia are chills, fever, prostration with occasional vomiting and diarrhea. The fulminating septicemia sets up rapidly, which can be fatal in many cases. The infection and fatality rate is very high, 40 to 60% among people with liver and gastric diseases and immunodeficiencies. Vibrio cholerae. Vibrio cholerae is best known as the cause of human cholera, contracted from polluted water, and evolutionarily 7 pandemics have been recorded due to Vibrio cholerae. In the 19th century, both epidemic and isolated cholera cases were recorded in US. Robert Koch isolated what is now known as the classical Vibrio cholerae biotype. This was responsible for the outbreaks of cholera until 1961. The Eltor biotype, first isolated in 1906 by Gottschalk from pilgrims bound for Mecca at the Eltor quarantine station in Sinai, Egypt, is responsible for the current or the 7th pandemic. This started in Celebes, Sulawesi in the Indonesian 1961, reached Africa in 1970 and the Americas in 1991. So slowly this spread from its area where it was first spotted to Indonesia, to Africa, to the Americas. This was recognized in the 1930s that both the biotypes are agglutinated by a single antiserum designated as 01. The other strains of Vibrio cholerae do not react with this antiserum 01 and they are termed as non-agglutinable or more correctly as non-01 strains, although some do produce cholera toxin. So, identification of the strain is as 01 and non-01. This is because of the agglutination, which it is agglutination response which shows to the antiserum. But many strains do produce what we call as the cholera toxin. In 1992, a new serotype 0139 was associated with epidemic cholera in India and Bangladesh, and this has also been isolated from cholera patients in Thailand. So a new serotype has latest come into picture in 1992, which is called as 0139. So you get cholera, as you know, because of contaminated water, and that you always drink only boiled water, don't take food which is raw contaminated food. Usually people will be carriers. So even if there is no symptom, if the person is a carrier, then that person can pass on the organism to the food. The strains in the 01 are associated with the epidemic cholera. This serotype is further characterized by biotype and serotype. Cholera epidemics worldwide are associated with the Eltor biotype and either the Inaba or the Ogawa serotype. Both the types are sensitive to heat and are killed by temperature used for cooking. So this cholera toxin, which is synthesized by either the Eltor biotype and the Inaba or the Ogawa serotype, they are sensitive to heat. So completely cooked food will not contain cholera toxin. Organism is sensitive to heat, and also the cholera toxin would also be killed. Organism would be killed. So there is no chance of it synthesizing the toxin, which in turn will contaminate the food and you consuming the food if the food is properly cooked. The non-01 serotypes do not agglutinate with the antibodies which are prepared against the 01 antigens. Similarly, the non-01 serotypes, similar to the 01 serotypes, they are not sensitive to trimethoprim, sulfamethoxazole with furanol. So you have sensitivities to the antibiotics prescribed also between the non-01 and the 01 serotypes. The disease results from the ingestion of infective doses of Vibrio cholerae cells through food and water contaminated with the feces of the human suffering from the disease. So this is a fecal-oral contamination of the food. You consume a food which has been contaminated with the Vibrio. Chronic carriers are rare and may not be important in large epidemics, and the long-term reservoir normally is the marine environment. The toxin of the 01 serotype is heat labile, that is, it is sensitive to heat. The 85 kilodalton cytotoxic protein with 2 functional units. Cytotoxic meaning it can lyse the cells, heat labile, it is heat sensitive. So the toxin created by Vibrio cholera 01, it is labile, it is a 85 kilodalton cytotoxic protein. It has 2 functional subunits, A and B. The active A subunit stimulates adenylyl cyclase in the intestinal epithelial cells. So each subunit of this cholera toxin has specified roles. The A subunit stimulates adenylyl cyclase, which is a signal molecule in the intestinal epithelial cells. This stimulation of adenylyl cyclase causes massive secretion of water along with chloride, potassium and bicarbonate in the lining of the intestine. So this will stimulate from inside the cytoplasm or lumen of the intestinal cells. Secretion of water will happen from inside to the intestine along with the ions, secretion of the ions chloride, potassium and bicarbonate. The non-010 serotype produces a cytotoxin and a hemolysin. So 01 produces a cytotoxin, the non-01 produces a cytotoxin and a hemolysin. Following the ingestion of the Vibrio cholerae cells in sufficient numbers, the cells colonize the small intestine, they multiply rapidly and produce the toxin. And when the cells die and lyse, the toxins are released into the intestine. So here the Vibrio cholerae cells colonize the small intestine and they multiply rapidly and they synthesize the toxins. And when these cells die, the toxins are released in the intestine. So this is how the cholera toxin, this is the Vibrio cholerae, this is the cholera toxin release, this goes and binds to this receptor here, gets inside, and this is stimulated, this is a G protein couple, this will go stimulate this. This in turn stimulates the adenylyl cyclase, which in turn stimulates cyclic AMP, which is a signal molecule. The stimulation of cyclic AMP goes and opens a transmembrane receptor, which in turn will send out of this lumen chlorine ions, then you have the water coming out, sodium ions, potassium ions, etc., showing what you now call as the typical symptoms of diarrhea. The symptoms include sudden onset of profuse watery diarrhea and vomiting. The loss of fluids result in dehydration. Other symptoms in severe cases are painful muscular cramps and clouded mental status. Many infected people do not show any symptom or may have only mild to moderate diarrhea. And normally, treatment consists of rapid replacement of fluids along with electrolytes and administration of proper antibiotics. Food can serve as the primary source of Vibrio cholerae if it is contaminated directly with human fecus from the patient or previously contaminated water. The handling of food by a person suffering from the disease can also contaminate the food because of poor personal hygiene. Food originating from natural reservoirs of the causative organism can be contaminated and this can spread cholera. The natural reservoirs include marine and brackish water environment. Unsafe water and poor sanitation is the warning normally given. Then properly take personal hygiene, proper sanitation, wash hands after restroom use, drink only boiled water, eat only completely cooked food, don't eat raw or unsafe. These are the precautions which are prescribed in endemic areas where there is an incidence of cholera. Isolation of Vibrio cholerae from a sample is achieved by an initial pre-enrichment in alkaline peptone water, followed by streaking on a selective agar plate, such as either the thiosulfate citrate bile salt with sucrose agar. Suspected colonies which look yellow are biochemically and serologically tested for confirmation. Toxin is detection by the immunoassay or bioassay. The toxin gene can be identified by PCR. So, if you suspect Vibrio cholera, you have to enrich the sample which you have taken from the food or from the patient in alkaline peptone water and streak on a selective agar plate. The suspected colonies would then be tested for biochemical reactions and then you serologically test it for confirmation. The presence of toxin in the food can be detected by immunoassay and the toxin gene can be identified by PCR using specific primers. So, if you look into the 3-year data, for example, this is a data which is prevalent to Gujarat and India. In the 3 years 2016, 2017, 2018, you had cases of diarrhea, typhoid, hepatitis and cholera. All these are water, if you see all these are waterborne diseases, mainly by the contamination of sewage water with drinking potable water. So, normally if you are not sure of the hygiene, please avoid food and water outside, and especially during travel, carry your own bottled water, and do not, if you are going trekking, do not take water from stream, river, lake if you are not sure about the source. And even inside a house, the water tank has to be cleaned periodically, and make sure that washing of the hands before and after consuming food or work as a habit. So, this is a normal health alert which is given in any area where there is a precedence of waterborne diseases. The other type of organisms which are prone to cause enteric diseases are enteric viruses. Unlike bacteria, not much is known about the incidence of viruses in food. We have more knowledge about bacteria and fungi. The first reason for this is viruses are obligate parasites. Okay, so they do not grow on culture media as do bacteria and fungus. Viruses require a live cell for their growth and replication, whereas bacteria and fungi can be cultured on culture media. And the usual methods for viral culture depends upon tissue culture and the chick embryo techniques. Second, since the viruses do not replicate in the food, their numbers may be expected to be low relative to bacteria, and extraction and concentration methods are necessary for the recovery. So if you suspect that the food has been contaminated with an enteric virus, as we discussed earlier, pathogenic bacteria incidence itself is very low in a food when compared to the normal microbiota which is seen. So even for identification of the pathogenic organism, bacteria, we require pre-enrichment. And if you take the numbers of viruses, they would be even more less. So you require extraction of these viruses and then concentration of these viruses for the recovery and then identification. And third and the most important, laboratory virological techniques are not practiced in many food microbiology, more of important are the bacteria and the fungi which cause food poisonings. And finally, not all viruses of potential interest to food microbiologists can be cultured by the existing methods. For example, Norwalk virus, which is very important as a reason for foodborne illnesses, it cannot be cultured normally in the lab by the existing method. So if you cannot culture it in the lab, how will you find out how to detect it, and what about how it causes the disease? What is the method of detection? So this is actually the negative aspect regarding. That is why we don't get much details about foodborne viruses. Foodborne viral infections can occur only from enteric pathogenic human viruses. Before the 1940s, polioviruses transmitted through raw milk were considered the only virus which was involved in foodborne viral infection. Recently, hepatitis A, Norwalk-like viruses, also called as the small round structured viruses SRVS, the rotaviruses, and the non-A, non-B hepatitis viruses have been associated with foodborne infections in different parts of the world. Among them, hepatitis A and the Norwalk-like viruses are more predominant, and both the hepatitis A virus as well as the NLVs are small RNA viruses, meaning their genetic material is ribonucleic acid, not DNA. Hepatitis A virus can be grown in cell cultures, and this can be used to produce vaccines. And the methods so far which have been tried to grow NLVs in cell cultures have not been successful, and normally their presence in the patient's stools is detected by enzyme-linked immunosorbent assay or the ELISA. Broadly speaking, there are two main types of pathogenic enteric viruses which differ in the target tissues. The viruses enter the body via the gut, that is via the food, but the gastroenteritis viruses remain confined in the gut itself, whereas others like the polio and the hepatitis viruses, they cause illness once they have migrated to the other organs. So, enteric viruses enter the gut, but then those viruses which show gastroenteritis remain in the gut itself. But the other viruses like your hepatitis A virus, they have a different place where they multiply. So, they enter via the gut, then migrate to that particular tissue, and then after reaching that tissue, they will start their illnesses. Hepatitis there are A, B, C and D types of hepatitis are there. A, B, C, D, E types of hepatitis. Normally A, B, D and E. This is an estimated case. You have so many estimated cases in the year. Normally contaminated food and water is the reason for the spread of hepatitis A. The symptoms of hepatitis A is pain in the abdomen, fever, causes jaundice, then digestion disorders, nausea, vomiting, feeling weak. The final manifestation would be dark urine, showing abnormality of the liver. Enteric viruses can cause infection at a considerably very low dose level. And if you look at hepatitis A as a case, following the ingestion of the A viruses through contaminated food, an individual may or may not develop symptoms. In infected individuals, there is an incubation period. The symptoms occur after 4 weeks, within a range of 2 to 7 weeks. So you might have consumed the food some 1 month to 1.5 months previously, but for you to fall ill with the disease, it takes close to 2 to 7 weeks. The general symptoms are fever, malaise, nausea, vomiting, abdominal discomfort and inflammation of the liver, which may follow with jaundice. And the symptoms last for 1 to 2 weeks or longer. The viruses are generally shed via fecus during the last half of the incubation period. Food contaminated with fecal matter of the infected people directly, that is the food handlers, or indirectly via sewage and polluted water, is the main source of both hepatitis A as well as the NLB outbreaks. Infected food handlers, even without the symptoms, can contaminate ready-to-eat food with fecal matter. The agent of enterically transmitted non-A non-B hepatitis has now been designated as hepatitis E virus, and molecular biology studies indicate that it is a calyce-like particle with an unusual RNA structure. This is also transmitted by the fecal-oral route and produces illness after an incubation period of 40 days. Vegetables, especially salads, can be contaminated with polluted water. Shellfish, oysters, clams, mussels, harvested in water contaminated with or polluted with sewage water and eaten raw or not properly cooked. Both have been implicated in the outbreaks of both types of viruses, and shellfish serve as a reservoir for the viruses. Two major preventive methods of foodborne viral infections are there to kill the viruses in contaminated food and to adopt good sanitation and personal hygiene habits to control contamination. Heat treatment of the food like pasteurization is enough to kill the viruses. Both for the Vibrio cholera diseases as well as for the foodborne viral enteric diseases, the basic precautions you have to take would be: 1. The food handlers, even if they are asymptomatic, have to be taught sanitization and personal hygiene techniques. 2. The food has to be properly cooked and stored properly. It is better to eat cooked food rather than the raw, especially seafood and salads. And in endemic areas, it is always better to be safe. The questions for this session would be: what precautions will you take to avoid the diseases which are caused by foodborne viruses? And how will you identify the foodborne viruses?